Welcome to the Rozovsky Group Recruitment Poster. I'm Sharon Rozovsky, and I will be introducing our team today. You can see us here in the lab or in a Zoom meeting. We are a biochemistry and biophysics group, and we integrate multiple tools to study the structure and function of proteins, from structural biology to that of cellular assays and imaging, to the use of biological mass spectrometry and enzymatic assays and enzymes inhibition. Our main focus is how does a cell protect itself from reactive chemicals that can damage membranes, proteins, and nucleic acid. This type of damage accumulates over time and leads to neurodegenerative diseases, to cancer, and to aging. The cell has several defense mechanisms that needs to be integrated and fine-tuned to control the level of stress in the cell. And much of it is done by a group of enzymes that have the element selenium in their covalent structure. This group of proteins, called selenium proteins, uses selenium because it's chemically very reactive. They're all enzymes, and they use the selenium as part of the way to enhance the chemistry available to remove those reactive chemical species. At the same time, they also fine-tune and control the level of cellular response to many different types of stress. And my group is interested in understanding how the selenium protein both sense and prevent cellular damage under stress. I want to tell you a little bit about the research direction in the group in the last 2 or 3 years. And we starting with a new mechanism that we discovered by which proteins can regulate their own interaction with protein partners and activity. Here we see a membrane selenium protein called selenium protein K. It is present in many different types of protein complexes and has different cellular roles. It is part of the cellular machinery that detect whether proteins are functioning correctly. Are they properly folded? Are they modified correctly? Do they have the disulfide bonds or sugar groups that they need? Are they in the right location? Do they have the right protein partners? And if not, this cellular machinery will degrade them to prevent processes such as aggregation or possibly misfunction. Another complex and function for selenium protein K is that of lipidation of proteins. By attaching lipid to proteins, selenium K can move them into the membrane, relocate them to membranes, and it could also change their lifetime. It is also present in lipid droplets, which are organelles that control lipid synthesis. And in here, selenium protein K has an immune role. This is not well understood, but it probably couples the immune response to invasion by viruses or bacteria to regulation of lipid synthesis. Selenium protein K is unique in that it's an intrinsically disordered protein. It doesn't have a stable three-dimensional structure. But at the same time, it's probably an enzyme because any protein that has selenium is an enzyme. How could it be an enzyme and yet not have a stable three-dimensional structure? What is the role of the selenium cysteine? What we found during our investigation is that selenium protein K is found in human cells in many different forms. The bands that you see in here represent the full-length selenium protein K, but also shorter forms. And after investigating it in great length, we found that that cleavage is from selenium K itself. It can cleave its own peptide bond in 2 locations. Those locations are protein hubs, that is, these are sites where selenium K is interfacing with other protein partners. And in addition to regulating its own interaction with protein partners, it also releases a segment with a selenium cysteine, that is, it's terminating its own enzymatic activity. This self-cleavage has not been reported before for an intrinsically disordered protein. And we want to understand what is the mechanism, but also what are the biological roles. In addition to being able to cleave its own peptide bond and regulating the segment with selenium cysteine and the segments with protein interactions, we found that selenium K can cleave protein partners. So its activity is not restricted only to itself, but extends to other protein. And we are currently investigating what proteins does selenium protein K is capable of cleaving, and what is the biological role of that cleavage. And we are fascinated by the connection between that cleavage and the immune response of the cells to invasion. That leads me to the second research thrust in our group currently, and that is, what's the relation between selenium and the progression of COVID-19? It turned out that selenium is very tightly regulated with the recovery from COVID-19. And that's because there are several selenium proteins that directly interact with the coronavirus proteins. These are selenium protein K that we already introduced, as well as a second membrane protein called selenium protein S. Selenium protein S is part of the machinery that controls protein quality, that is a machinery that detects whether they're damaged or properly functioning. But it's also involved in trafficking of components in the cell, trafficking vesicles. It's tightly connected to cardiovascular health. It's connected to signaling pathways in the cell. And it is taken by the coronavirus replication complex. So it was reported that it is connected to one of the coronavirus proteins that is part of the replication complex. And indeed, we have found in our group that it is capable of integrating with the entire replication complex using its membrane segments. What we currently are studying is whether it influences the activity of the coronavirus replication complex, whether it is responsible for linking the coronavirus replication complex to membranes. What is the connection between selenium protein S and cardiovascular health and the viral progression? What is the connection in those signaling pathways which are part of the immune response? So this is all currently part of our active research. In addition, we found that selenium protein K is also interfacing with with coronavirus proteins. These are membrane-bound coronavirus proteins. And it appears that there's a relation between those complexes that forms and possibly interaction between selenium protein S and selenium protein K itself. And so in a nutshell, we want to understand what is the role of recruiting those selenium proteins to the coronavirus. Is it important only for replication of the virus or also for its ability to take over membranes? And whether these are potentially roles that we can understand and possibly target when looking for therapies for COVID-19. And so I want to encourage you to contact us if you have any questions. You feel free to reach out for any members of our team or to myself, and we'll be happy to discuss in more length any one of those projects and other research opportunities within a group. And I hope you enjoy this recruitment day, and I'm looking forward to talking to you in person.